Humans, are unisexual. They have a primary male or a female sex organs, and these organs are known as gonads. Gonads produce gametes and also the sex hormones. The male gamete is the sperm and the female gamete is produced from ovary and is known as the ovum. Humans are viviparous and the fertilization is internal. Human reproduction involves a series of events, and we can divide these into 6 steps. Step 1 is gametogenesis, or the formation of male and the female gametes. Step 2 is transfer of male gametes to the female genital tract, and this is known as insemination. Step 3 is fertilization or fusion of male and female gametes. The zygote starts to divide and forms a ball-like structure known as blastocyst. This blastocyst gets anchored or fixed in the female uterus where it develops. This process of fixing of blastocyst in the uterus forms the next step known as implantation. And finally, the development of embryo or gestation occurs. In humans, gestation period is 280 days. This is followed by parturition or delivery of the child. So in this lesson, we'll be doing all these events in detail and also a comparison of the male and female reproductive process. Our goals for this session are a detailed study of the organs that form the male reproductive system. We'll also try to cover the spermatogenesis or the formation of sperms. Now let's begin with the male reproductive system. Human male has 2 gonads, that is a pair of testes. These produce the male gamete or the sperms and the male hormones, that is androgens. In a male fetus, testes are formed in the abdomen. By birth, they descend into a separate sac outside the body, and this sac is called scrotum. Sperm production requires a temperature of about 33 degrees Celsius. Now you know that our body temperature is 37 degrees Celsius. So testes need to be outside the body at a lower temperature for successful sperm production. Now here are the structural features of testes. They are oval, 4 to 5 centimeters in length, 2 to 3 centimeters in width. They are covered by a dense covering. Inside they have 250 compartments or the testicular lobes, and they produce the male hormones, the androgens. Now we just studied that testes have 250 compartments. The sperm forming and the hormone making cells are present inside these testicular compartments. Each of the testicular lobe has 1 to 3 highly coiled seminiferous tubules. These seminiferous tubules produce sperms, that is they contain spermatogonia or the male germ cells. They also contain another set of cells known as Sertoli or nurse cells, which provide nutrition to the germ cells. The region in between the seminiferous tubules is known as interstitial spaces. It contains small blood vessels and interstitial cells or the Leydig cells. Leydig cells synthesize and secrete testicular hormones called androgens. The most important androgen is testosterone. This controls the development of sexual characters and spermatogenesis. Now the function of testis is spermatogenesis and secretion of testosterone. Now let's study about the accessory ducts. Seminiferous tubules you just studied are inside the testis. These seminiferous tubules open into a network of tubules known as reed testis, which further leads into vasa efferentia. These two ducts are inside the testis, collecting material from different seminiferous tubules. Next, the vasa efferentia leaves the testis and opens into epididymis on the posterior surface of each testis. Now, the main function of epididymis is conduction of sperms by peristalsis. You recall peristalsis? It was occurring in your digestive tract to push the food downward. It is coordinated contraction and relaxation of muscles. Epididymis also helps in storage, nutrition and physiological maturation of sperms by removing certain decapacitation factors. Epididymis leads into vas deferens that ascends to the abdomen and loops around the urinary bladder. It receives a duct from the seminal vesicle and opens into urethra as the ejaculatory duct. You have to remember the sequence of these accessory ducts for proper understanding of the passage of sperms. Now the male accessory glands. The first one is seminal vesicle. Its secretions comprise 60 to 70% of semen, mainly formed of fructose, citrate, several proteins and prostaglandins. They activate spermatozoa and help in fusion of gametes. Second one is prostrate gland. It produces alkaline secretions in urethra. These secretions contain lipids, small amount of citric acid, bicarbonate ions and some enzymes. Example, fibrinolysin. It forms 20% of the semen. The main functions are it activates sperms, provides nutrition to sperms, neutralizes the acidity of urine and adjusts vaginal pH. The third gland is a pair of bulbourethral glands which secretes mucus-like alkaline substance that provides lubrication. Secretion of these glands constitute the seminal plasma, which is rich in fructose, calcium and certain other enzymes. It keeps the sperms energy rich so that they can move. Thus, the major function of male reproductive system include spermatogenesis, secretion of male hormone that is testosterone, transfer of sperms to female reproductive system for fertilization of ovum. Now, what is spermatogenesis? Spermatogenesis is formation of sperms. Formation of haploid and functional male gametes, that is spermatozoa, from diploid reproductive cells, that is spermatogonia, present in the testis of male organism. It occurs in testis, begins at puberty and is a continuous process completed in 74 days. Now, spermatogenesis is divided into two phases: formation of spermatids and spermiogenesis. Let's focus on formation of spermatids first. This involves 4 steps. The first step is the multiplicative or mitotic phase, where the spermatogonia will divide to increase its number. Spermatogonia are present on the inside wall of seminiferous tubules. They keep on dividing. Their chromosome number is 46. The second step is the growth phase. Here, some of the diploid spermatogonia, now known as primary spermatocytes, will undergo meiotic division, leading to formation of primary spermatocytes. These primary spermatocytes will complete their first reduction division, leading to formation of two equal haploid cells known as secondary spermatocytes. Then these secondary spermatocytes will undergo the second meiotic division to form spermatids. So, spermatogonia undergo mitotic division. Some of these spermatogonia, now known as primary spermatocytes, will undergo meiosis 1 to form secondary spermatocytes, and then secondary spermatocytes will undergo meiosis 2 to form spermatids. Spermatids are non-motile. Now the next phase, that is spermiogenesis. The spermatids are formed. These spermatids we just studied are non-motile, so they have to be converted into spermatozoa, which is motile, and this process of conversion of spermatids to spermatozoa is known as spermiogenesis. The main aim of spermiogenesis is to increase sperm motility by reducing weight and development of some locomotory structure. After spermiogenesis, the sperm heads become embedded in Sertoli cells, which provides nourishment to them. Mature spermatozoa are finally released from seminiferous tubule by a process known as spermiation. Now, let's revise the process once again. You have spermatogonia in the testis. The spermatogonia contain primary spermatocytes, which are 2N. The primary spermatocytes will undergo meiosis 1, secondary spermatocytes, haploid, meiosis 2, spermatids, which are non-motile. Spermiogenesis leads to formation of spermatozoa, which are motile, and then spermiation, or the release of sperms. Now let's study the structure of human sperm. Now, this is an interesting structure in the human sperm, that is acrosome. It is formed from a part of the Golgi body of the spermatid. It is filled with enzymes that help fertilization of the ovum. The lytic enzyme present here is known as hyaluronidase, which is present in acrosome, and this is responsible for penetration of ovum during sperm entry. The middle piece has many mitochondria which produce energy required for the movement of tail that causes sperm motility. Sperm motility is essential for fertilization because it will help the sperm reach the ovum. Tail is the longest part of sperm. It exhibits lashing movements so that the sperm can be pushed towards the ovum. The fertility of a human male is dependent upon three characteristics of sperm. First is motility. The sperm should be motile enough to reach the ovum. The second is number. The human male ejaculates about 200 to 300 million sperms. For normal fertility, at least 60% sperms must have normal shape and size, and at least 40% must have normal motility. The third factor is viability. Viability of human sperm should be 24 hours for fertility. Now the control of spermatogenesis, a very important topic. Spermatogenesis starts at the age of puberty. The controlling factor for the start of spermatogenesis is increase in gonadotrophin releasing hormone, GnRH, gonadotrophin releasing hormone, which is secreted from hypothalamus. This acts on anterior pituitary gland. Now, high level of gonadotrophin releasing hormone stimulates release of two gonadotrophins from the pituitary gland, that is LH and FSH. LH stands for luteinizing hormone and FSH stands for follicle stimulating hormone. The LH acts on leading cells and FSH acts on Sertoli cells. LH stimulates the secretion and synthesis of androgens, which stimulates spermatogenesis, and FSH causes the Sertoli cells to secrete some factors which leads to spermiogenesis. We come to the end of this session. Here are a few questions for your revision. Try solving them. Draw the diagrams and flowcharts. These will help you develop a better understanding of the topic. Thank you. Bye-bye.